Okay, let's get started. Hi, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as chair of biostatistics and bioinformatics at Roswell Park Comprehensive Cancer Center. So a few points about today's seminar. In supporting the next generation of cancer researchers, today's Cancer Moonshot seminar will feature research from talented early career scientists. These are abstract-driven talks nominated by Cancer Moonshot investigators and selected by NCI staff. A few bookkeeping items. Questions will be responded to at the end of the second presentation. Please enter your questions in the Zoom Q&A box. The audience can prioritize the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions are also provided in the chat box. The recording of today's webinar will be available a few weeks after today's date. With that, I'd like to introduce our moderator, Dr. Evan Weber, an assistant professor at the University of Pennsylvania Perelman School of Medicine and head of a research lab at Children's Hospital of Philadelphia. Dr. Weber's research is focused on enhancing human CAR T-cell therapies for cancer and improving T-cell fitness. The various approaches, including pharmacologic, gene editing, and bioengineering methods, he and his team aim at identifying just T-cell intrinsic pathways to optimize CAR T-cell function. Dr. Weber's research aims to uncover molecular programs driving human CAR T-cell function and dysfunction, identifying new targets for therapeutic intervention, and developing universal strategies to improve CAR T-cell efficiency in cancer patients. Dr. Weber.
Thanks so much for the introduction. It's very exciting to be here. Having been a presenter of the series a few years ago, it's great to be moderating and to be able to introduce two outstanding investigators. So first up, we have Dr. Chris Bossi, my colleague here at CHOP. He's a physician scientist and an assistant professor in the Department of Pediatrics at the Perelman School of Medicine at UPenn and leads a research lab in the Division of Oncology at CHOP. Chris trained in the laboratory of Dr. John Maris, also at CHOP, where he studied functional genomics and translational science and really developed a passion for improving treatment options for children with high-risk neuroblastoma. Chris has received numerous awards for his work, including a KO8 from the NCI, Alex's Lemonade Stand Foundation A Award, and Hyundai Hope on Wheels Scholar Award, among many others. Chris's laboratory is focused on functionally validating cell surface molecules that are differentially expressed on neuroblastoma and developing immunotherapies like CAR T cells to target these molecules and then translate them to the clinic. Today, Chris is going to be speaking about one important neuroblastoma marker called GPC2 that he has worked on for many years, and also introduce novel therapeutic approaches targeting GPC2 positive neuroblastoma. So Chris, thanks for joining us. Looking forward to your talk.
Thank you, Evan. You guys see the slides?
Yes, we do.
Well, thank you very much for the nice introduction, the invitation, and it's really a real pleasure to give a talk in this forum, and a pleasure to have Evan now at CHOP, and hope I can convince him to work with me a little more. So as Evan mentioned, I want to talk a little bit about work we've been doing in neuroblastoma and focusing on this molecule GPC2, and it's a project that I started as a postdoc in John Maris's lab, and now I was studying in the independent lab. And just to get everyone up to speed, I'm sure there's non-pediatric oncologists on this call. So neuroblastoma is a disease of the peripheral nervous system. The so-called high-risk phenotype really does quite poorly clinically, which I'll show you in a minute. And there's been a lot of excitement recently targeting this sugar called GD2, both with a chimeric monoclonal antibody and more recently with GD2 CAR T cells. And really, I think has proven that neuroblastoma can be targeted immunotherapeutically. But really, when I started this project, and still today, there's really few other validated targets that in this cancer. And so this is a little bit out of date, but still accurate in terms of the prognosis event-free survival for kids with neuroblastoma. And you can see here in this bottom line here in yellow, these kids get anywhere from 18 months to 2 years of really intense chemoradiotherapy and still have less than 50% chance of surviving. So obviously you need to do much better in the clinic. And this is a patient here on the right. It's an MIBG scan. Actually, a patient I diagnosed as a young attending, and this is MIBG scan. All these white areas are signs of or spots of disease. You can see how widely disseminated neuroblastoma can be at the time of diagnosis. And so we were unhappy with these data, and obviously I wanted to do better. So we about a decade ago, which is sort of sad to think about, we started a study where we used RNA sequencing data to find really find genes that were differentially expressed in tumors not found in normal tissues. And it's all been published, so I'll go through it quickly. But we basically use target neuroblastoma data against GTEx normal data and found a list of genes that were differentially expressed but highly expressed and thought to be on the cell surface. And quickly, this molecule GPC2 or Glypican-2 shown here on the right with high tumor expression, very limited to no normal tissue expression, became really our favorite molecule and really what I built my lab around. And I'll just say that we validated this all at the protein level, this high level of differential expression. We didn't know anything about GPC2 at the time, and actually really true of all the entire list of genes. So there's a family, it's one of a family of 6 Glypicans, and they're all sit on the cell surface, GPI linked to the extracellular surface. And so, really perfect for immunotherapy. Everything's in theory targetable. And to get the core protein together with different sugar side chains, depending on the Glypican, they're thought to act as these signaling co-receptors, and they bring ligands and receptors together and drive processes that are important both in development, which will become important in a minute, and in cancers. And we don't quite know for sure what GPC2 does, but there's some hypotheses I've listed here. I trained as a genomicist, so we think a lot about where these genes are encoded, and GPC2 is on chromosome 7q, which is somatically gained actually in a fair number of neuroblastomas, although we don't talk much about that and sort of the neuroblastoma segmental chromosomal aberration literature, but it is quite frequently gained and plays a role into its differential expression. And I think what got me really excited about GPC2 was the fact that this sister molecule, GPC3, was very heavily targeted and commonly targeted in hepatocellular carcinomas and some pediatric liver tumors. So really proved that you could target this family of proteins. So GPC2's, it's interesting developmentally, it's expressed very highly in the developing nervous system. Neuroblastoma is a peripheral nervous system disease. So it's not a brain tumor, it's a peripheral nervous system disease. But nonetheless, GPC2 is high in developing nervous system tissue and then really is lost at birth. This red line indicates birth for all these different mammalian tissues. This is for humans. You see the same trend in all mammals. And again, you see very limited normal sort of adult mature tissue expression. And I'll come back to this, but this is Anna Maria Gadish, who's a postdoc who joined my lab, unfortunately, during the pandemic. And she got very interested in this notion that Glypicans were very high on extracellular vesicles or exosomes. And this will become important in the last part of the talk. And it wasn't, we had thought about this for a while, and it wasn't until she came to the lab that she developed the techniques to do via differential centrifugation to isolate these small vesicles you can see here in the lower left, these 100 to 200 nanometer vesicles that are secreted by all cells, actually very commonly secreted by cancer cells. And then you can put these, link these to beads and do normal flow cytometry. You can see high levels of GPC2 on vesicles, either from the blood of tumor-bearing mice or the cell lines or PDXs themselves. And most importantly, when she looked in the blood of children with neuroblastoma versus healthy normal controls, you can see selectively they see GPC2 on vesicles, extracellular vesicles from kids with neuroblastoma. You can see the raw flow on the right. So really suggesting it's a very neuroblastoma specific thing, really suggesting it's coming from the tumor, and something that I'm going to come back to in a minute. But we also think it might be a very important peripheral diagnostic tool for kids with this disease. And we're lucky, and through both cancer moonshot funding we've had and a Stamp the Cancer team, we collaborate with a group that used to be at the NCI, now it's the University of Pittsburgh, the Dimitrov lab, now run by Wei Li, who are experts in finding antibodies. And of course, to target any molecule with any immunotherapy, you need a good antibody. And so we were lucky, and this has been published, so I'll go through it quickly. We're lucky to find several good GPC2 antibodies. Our favorite is this D3 clone, and this is a picture of the D3 Fab actually crystallized with the GPC2 here on the left in brown. This is work done by John Philippe Julian's group in Toronto. And the important thing is it really allowed us to really pin down the binding amino acids on the protein. And you can see here that both the heavy and light chain bind these sort of conformational epitopes. So there's N-terminal amino acids and C-terminal amino acids that come together conformationally. And the importance of this, we think, at least at the RNA level, is tumors express this canonical exon 1 to 10 GPC2, and the few normal tissues that have a little bit of expression have a N-terminally truncated, really missing half of the D3 epitope. So we think this will enhance the therapeutic index even further than what we get from just the differential expression alone. The antibody binds mouse and human GPC2 the same, and importantly, doesn't bind anything else in the human cell surface. And so, as I've mentioned, I've spent the better part of my independent lab, again, sort of scary to think about, but targeting this one molecule. And the first thing we focused on doing, and there's certain just practical reasons for that, was making an antibody drug conjugate. And I won't talk very much about that today because we're doing a lot more work in the CAR T cell field. But the first thing we made was from this D3 human IgG was this PBD, which is a pyrrolobenzodiazepine dimer. It's a very potent DNA intercalating agent. This is a cleavable, enzymatically cleavable linker. So we made this and actually showed, it's been published very potently that it's safe and potent in neuroblastoma and actually other GPC2 expressing cancers. And a lot of that published work is here. And we've worked now working with a third company to try to get one of these an ADC targeting GPC to the clinic. So hopefully much more to come there. I do think for solid tumors, ADCs will be a very important therapy in the clinic, and whether they're more or less effective than CAR T cells, we'll have to see. But I think building off that and using the same antibody, we've now made several CAR T cells, and I think probably most people on this call understand what a CAR T cell is. But just briefly, we've really focused on making second generation CARs where you have the antibody. We've largely favored using a CD28 TM and with either 4-1BB or CD28 and a CD3 zeta signaling domain. And I'm not going to go through the CAR manufacturing process, but I think most people understand that. And we've optimized these a lot, again, in some published data. But what I wanted to show today was some published data that actually went into the IND that enabled us to launch a trial in the last few months here at CHOP. And we really focused on, again, based on a lot of this published optimization, 2 CAR constructs, one with both identical, except for one had a 4-1BB and the other had a CD28 TM, hypothesizing that the 4-1BB would give us better persistence, but not knowing, what the case would be with GPC2. We have a panel here with high and low GPC2 expressing cells, and you can see in this sort of real-time sensing assay, you kill very potently GPC2 positive cells here on the left, but then on the right, you see no killing with the GPC2 negative cells. That made sense. And then in terms of T cell activation, these are interferon gamma and IL-2 lyses. You get very, high levels of these cytokines in the supernate. And when you co-incubate these CARs with target cells with GPC2, but not when the cells don't have GPC2. So it's a very GPC2 specific T cell activation, killing, again, as one would expect. And then most importantly, we rely on PDXs in the lab, and this is a high GPC2 PDX, right there, 421X. You give one dose of 10 million CARs when these tumors become established in the flank of mice, about 200 millimeters cubed, and you see good regression versus this control PSMA non-targeting CAR. We quantified the tumor volumes. We took some of these mice off for full necropsies for toxicology studies for the IND, and you can see just how the tumor volumes differed at those time points. I will say, I won't show you the data, but there was no GPC2 CAR specific toxicities. The T cells widely traffic in NSG mice, but there's nothing unique to the GPC2 trafficking or toxicity. We, at some point along the way, decided the 4-1BB CAR was really the CAR we'd move to the clinic. So this is just a cell line xenograph showing, again, good efficacy and tolerability of the 4-1BB CAR alone versus a PSMA CAR. And so this and a lot of other data I'm not going to show you for time went into an IND that thankfully got approved by FDA last May and helped us launch a phase 1 first in human trial for GPC2 CARs. First, initially focused on relapse refractory persistent high-risk neuroblastoma patients with a goal of 30. We've now enrolled 3 patients, about to dose our 4th patient in the coming weeks. And it's just a standard 3+3 design, really focused on safety and feasibility and with some preliminary efficacy assessments. And so that's all been very exciting. And we're doing a number of things now in the lab to, trying to predict what mechanisms of relapse, trying to build off this initial trial, these initial data, a lot of which I won't have time to talk about. But what I really wanted to focus on in the second half of this talk is how Anna has used and really understood how these vesicles are interacting with CAR T cells. And so we're of time, I would have more time to talk about other things, but I think this is what I'm going to focus on. So Anna got very interested in understanding how EVs released from neuroblastoma cells either negatively or positively affect CAR T cells. So she made a suite of these EVs that have different amounts of GPC2. They basically phenocopy what's on the cell. CD81 is an EV marker. And what she saw very early was when you incubate these with CARs, the CARs here are labeled with DAPI, the T cells, and the green are the EVs. You can see that they when you have GPC2 on the EVs, they bind CAR T cells. They don't bind, and when there isn't GPC2, they don't bind the controls. You can see that by flow here. You have, again, you can quantify the green human T cells, and you can see that the more GPC2, the more EV, the more binding you have. And so that was interesting, but then we said, well, are they activating? Are they inhibiting? They seem to be activating this is ELISA after 24 hours of EV GPC CAR T cell incubation. You can see high levels of cytokines, whether you have a 400B or 28. You can see that by flow with CD69, the more EV GPC2, the more T cell activation. And then when you take these activated cells and put them on target cells, not surprisingly, they kill target cells better than if you didn't pre-incubate them or you incubate them with control EV. You can see, I think most impressively here in the 401BB thing in the middle with these are relatively resistant cell lines that, again, when you pre-incubate with EV, you can get enhanced killing. So what we really wanted to understand is, what happens in vivo. And there's ways you can modulate EV biosynthesis and in the literature, and people genetically deplete RAB27A, or there are drugs that inhibit this neutrosphingomyelinase 2, and you see the pathway here. And so Anna used CRISPR to knock out RAB27A. This, again, is a relatively resistant cell line. You can see lower levels of EVs. It's not zero, but it's significantly lower when you knock out this protein. And so then she did a pilot study where she took actually these cell lines and made xenografts, and you let these get to a pretty big size, 300 millimeters cubed, and then gave them CARs and followed with just intratumoral injection of GPC2 positive EVs. And this is a cell line that xenograph that we didn't expect to respond. And so you can see here in the blue, you can, starting at 300 millimeters cubed, you get limited efficacy of the CAR alone, but that's enhanced when you add GPC2 EVs into the tumor. And you can see this plotted out here. And I think, most interestingly to me was when you look at the number of activated T cells in the tumor, they're significantly higher when you have EVs present. So we wanted to show that a different way. And when you use this drug, this neutrosphingomyelinase, people have shown that this inhibits EV secretion in cells. It doesn't inhibit tumor growth, which was good. But that when you look at the EVs, whether it's in the blood or the tumor, you see lower levels of EVs in animals that got this drug. So Anna designed another study, again, here using the same cell line, but actually ones we've endogenously put in GPC2, but using a very low level. This is about 1/10 of the normal T cells we would give a mouse, and really just asking the question, in this setting, how important are the EVs? And this is a model that will respond well to CARs, at least CARs that we give, at the higher dose. And then she had cohorts where she had control CAR plus and minus drug, GPC2 CAR plus and minus drug, and then an arm where she added back EVs after giving drug. And here she saw a very similar thing to the last study. The only arms where she saw efficacy was arms where she had the EVs present. So either the car alone or the arm where she gave back the EVs, really suggesting that in the setting of high GPC2, the EVs are still very important with this low level of CAR T cells. You see the same trend in tumor volumes when you plot these out. You see the same trend in survival. Again, the arms that have EVs present were the ones that are the most efficacious, and you see the same trend in the T cell trafficking to the tumor. And so I'm almost up out of time, but I'll show this one last slide. And this is an ongoing study. We got really excited about this. So this is really interesting. So she built some EVs, and I'm not going to go into how we did this, but she built some EVs to try to understand where the EV should be once, should it be in the tumor, should it be in the blood? Is this a way to vaccinate against CARs? So she made EVs with albumin binding that should, in theory, live in the blood longer. These normally have a very short half-life. And then she made others where there was a GD2 antibody on the EV where, the hypothesis this would home to the tumor. And then here we treated SKNAS with GD2 or GD3 synthase that gives you high levels of the sugar on the surface, thinking this would enhance targeting of this GD2 EVs. And what we saw was actually really good efficacy of all the EV groups. These are now we're giving these IV twice a week for 2 weeks and then weekly thereafter. So these get IV EVs, which is different than the prior study, and they all look very efficacious compared to the CAR alone here in blue. So we didn't really answer the question, where do they need to be? But I think answering the question, giving them IV is actually even better than giving them into the tumor. And when you look, actually, I think the most telling is when you look at the number of T cells in the blood, and the CAR alone, the T cells are basically gone by 20 days, but the EV, the cohorts with the EVs with the albumin have really a high number of T cells still present, and in this cohort. So a lot more to learn here, but I think it tells me that giving extratumoral antigen, whether it's in the form of EV or something else, is really important for solid tumors, which I think is not too surprising. So we're going to really try to build off these studies. And I'll just end with this one slide saying that, I talked about neuroblastoma, might give you a little bit of input on EVs, but our hope is really to get it to, and this is a slide I made for a grant recently, to all these different tumors that have GPC2 expression. So hopefully I've showed you this. I think my time is just up, but how excited we are about targeting GPC2, both with CARs, which I talked about, with ADCs, which I didn't have a talk about, and this whole idea that EVs in solid tumors may be really important for efficacy of immunotherapies. And again, I pointed out Anna, but just thanks to obviously my great group and great collaborators. And I only have a short period of time, so if you have other questions about other work, please do send me an email. So thanks very much.
Fantastic. Thanks, Chris. That was an amazing talk. And for those who have questions, please put them in the chat and we'll address them after our next speaker's talk. So
Sorry, Evan, just a quick, put them
go ahead.
put them in the Q&A box.
In the Q&A box, right? Thank you. All right, so while we're loading the next presentation, I will go ahead and introduce our next presenter. So I'm pleased to introduce Dr. Xiao Huang, who opened his lab in 2023 at Drexel University in the School of Biomedical Engineering, Science and Health Systems. Xiao trained at UCSF under the mentorship of Dr. Tejal Desai and Qi Tang, where he developed DNA scaffold-based biomaterial technology to modulate immune cell function, including CAR T cells targeting cancer. Xiao is the recipient of numerous accolades, including the prestigious Burroughs Wellcome Fund Rising Stars Award. So now at Drexel, Xiao's independent lab is developing novel engineering-based approaches to precisely control CAR T cell phenotypes, something that I'm very interested in as well, and also developing new platforms that integrate biomaterials, engineering, synthetic biology, and systems biology to improve immunotherapies and generate a deeper understanding of immunological processes. Today, he will be talking about some very cool work from his former lab and his new lab that leverages functionalized biomaterials for CAR T cell manufacturing and T cell fitness enhancement. So Dr. Huang, thanks for joining us and looking forward to your talk.
Thank you, Dr. Weber, for the wonderful introduction, and also thank you for IoTM for the opportunity to share my research findings on precision biomaterials and its use in T cells, CAR T cell modulation, and also point to exciting future directions. Actually, before I actually get to my research here today, I want to give brief introduction of myself and also my lab. So actually, in the past decade, 10 years, from my research journey, I have been working at the interfaces between materials and cell systems in terms of different therapeutic applications. So, and my lab, actually, as introduced, my lab just started last June, and time passed quickly, and I'm excited to have our function being coming up very soon. And today, as I introduced, my lab is really excited about using engineering tools, advanced engineering tools, to tackle the challenges of immunotherapy. And today I want to mainly talk about the work. I would say a transition work from my postdoc to my independent lab. Which is on the CAR T cell modulation. So, I mean, you may have heard many talks about this. I do not want to spend too much time talking about too extensively the background, but as you all know, it's actually very successful in treating the acute lymphoblastic leukemia, and then also have been caught many attentions in diseases beyond cancer, for example, the autoimmune diseases. So as promising, but also it's also has some challenges or limitations. So right now, I would say that from my learning of the field is like there's really a lack of cell persistence, and also this is coming with a very high manufacturing cost, and also there's of tumor toxicity and lack of the solid tumor efficacy as I envision this as I review these challenges. So for me, actually, I really interested in trying to tackle these challenges through engineering. So from my training of immunology, I start to think more and more about how naturally T cells are programmed in our body by antigen-presenting cells for clonal expansion. While also when T cells are exposed to targeting, for example, cancer or other pathogenic cells that we want to target. So there must be a difference that I actually reviewed from a lot of the literatures and also from my own work, that these two processes is actually very different, that can be linked to different cell phenotype and cell features, that a deeper understanding of this mechanism may be helpful for us to tackle the challenges listed here. So actually, the reason why I'm so excited about using engineering platform is because, as you know, engineering, materials engineering especially, we can actually control each single factors in a precision way to understand better about the biological process, and then even find translational solutions to them. So then really, so my early years of postdoc work, I was trying to build a good hammer for tackling the challenge. So as you all know, that on immune cells, there's a spectrum of different receptors. So they always acutely sense signals from extracellular space, and then to decide their phenotype. So really, how can we engage those receptors effectively? That really, if we want to use engineering platform, we must first tackle the challenge of how do we give those biological signals on the surface. So this motivated me to design this platform to tackle the challenge of the surface bioconjugation limitation. So previously, it's actually very difficult to get a good surface loading high density enough. But then I refer to this DNA synthetic short DNA scaffold based approach that just simply use DNA hybridization to functionize the biomaterials on the surface. So what this approach has been shown in our earlier publication is that we really achieved a very high density of proteins on the surface and almost reaching to the theoretical footprint limit of the surface. And also using DNA approach, we found that we can now do a precision control of the different signals that we functionize on the surface. So with this platform, so we actually implement in this CAR T cell manufacturing procedure as first of proof of concept. And as Dr. Bossi has introduced about this CAR T cell manufacturing, so T cells are getting out from the body and then engineered and then reinfused back. So during this, the Dynabeads is used to provide signals for the TCR and co-stim stimulation, which are very important to signal 1 and signal 2, while giving out to make T cell proliferate to manufacture them. And then, so here we use we mimic that strategy. We also give the anti-CD3 and anti-CD28 antibody similarly. But when we implement this precision control of the two signal, we found that correlate well perfectly with the cell expansion fold in the beginning. And also when we actually monitor the cell phenotypes of those expanded cells, we saw that certain ratio actually give us more of a memory-like cells, while the other ratio give us more of a effector-like cells. So we think this opens an interesting box that, by this precision engineering, we might be able to further control the T cell phenotype or fate. So which actually also excited me to try with the CAR antigen-specific stimulation. So here, this work is unpublished, which we were trying to functionize the particles with antigens that can be recognized, can bond to the chimeric antigen receptor and also give a co-stim signal on the surface. And then we tune the ratio. So we found that in vitro or ex vivo with this human CAR T cells, here is the example of EGFR CAR. So we found that when we tune the ratio of the antigen to the co-stim signal, we also see a correlation of cell expansion by that first round of stimulation. And then at a specific ratio that give us, like among 3 donors, the best cell proliferation. So, and also next, we were wondering, can we do repeated stimulations? So can the cells be continuously proliferating? The answer is yes, as you see in this graph, that the donors are correlated. And we see a very persistent and also very consistent cell expansion among different donors by this repeated stimulation when we give particles every 8 to 10 days. So with these features, we're also interested in a different type of second generation CAR, which is CD28 co-stim domain CAR. So here, the patterns also form similarly, but the addition of the co-stim does not seem to be quite important to give the best cell expansion for the first round of stimulation. And then that also point to something interesting about the requirement of the co-stim signal. And we also do this antigen specific without co-stim to see if the different CAR T cells from different donor can also respond to continuously proliferate. And then you can see like, yes, we can achieve that like beyond 100 days, they're still proliferating. The overall cell expansion fold for both types of the second generation CAR is massive. So this is like beyond 10 to the 12th fold of expansion. And also, I want to point out another interesting feature is with this donor, we name it donor Z, that it does not respond that robust compared to the other donors. If we give additional co-stim signal, actually that respond much better. So this is the donor Z in data. And also some other donors, donor D actually also is the same thing. If we give this co-stim, they respond a lot better to be continuously proliferate. So the next question we want to ask is that those massively expanded cells, where are they still good effector cells? So the answer is yes. We give this in vitro co-culture, give this K562, which is leukemia cell line, and then we on the x-axis is the cells that has gone through multiple rounds of S particle stimulation and the particle stimulation, and then the cells expanded massively, and then we control the E2T at defined ratios, and then try to see do we lose any effector function? The answer is clearly no. And also, what's interesting that after 3 or 5 rounds of expansion, they have gone like about 1 million folds of expansion. Their effector function is actually better than in the beginning. And also we test this in different cancer target, and then the trend is very similar. And when we also try to repeat challenge the CAR T cells, and then the conclusion here is that we do not observe any loss of cell function at all for our massively expanded cells. So next, actually, we refer to this technology of seahorse analysis to monitor the mitochondrial fitness. And also, consistently, those 3 or 5 rounds of particle stimulated cells, even though they expanded a lot, but also they even have somehow an augmented mitochondrial fitness. So in terms of spare respiration capacity measurement. So this is the function. And next, our question is, with this massively expanded cells, so is there any clonal enrichment? So to get that question, we must refer to the single-cell RNA-seq technology. And with this, by collaborating at UCSF with Jimmy Yi's lab, actually we use a state-of-the-art technology that we can actually sample all the from many different time points and combine all the donor together. And then as well, we can also coordinate the protein surface expression by the CySeq and the total transcriptomic analysis to make conclusions. And from the TCR repertoire analysis, you can see the data shows clearly there is a clonal enrichment among all 3 different donors. So, and then we see super clones that actually proliferate a lot more. And then next on this UMAP, due to the time, I would roughly go with the conclusion. So when we actually put it on this UMAP, we found that those unstimulated cells, before stimulation, they mostly sit on this red area, so on the top half. While expansion, they actually move downwards. And then by looking at this different cluster, actually cluster 5 among both RNA analysis and the protein level analysis, we tend to believe that 5 is more of an effector memory feature. So especially with the markers of IL-7 receptor, CD62L, and also interestingly, our particle stimulation did not really get a very high level of exhaustion marker, typical exhaustion marker expression. And this is the clusters that I mentioned, but this is just giving you another view of the data, which really shows that the cells we got expanded on cluster 5 and 6, they live for a long time, but there's two dramatic different cluster. One is more of effect memory. The other is more like I interpret it as exhaustion progenitors. And also interestingly, those cells are caught on the G1 cell cycle. So meaning like they actually rested faster to stay down G1 to wait for more stimulations. And also another point I want to mention is with this a repeated stimulation, we also monitor the exhaustion associated markers. We do not see a quick increased level of exhaustion marker expression at all. So as also I mentioned in my introduction slide that however you activate the T cells really matter. This is the data we saw. If we activate the CAR T cells with K5C2 cells, we quickly get the cells to be apoptotic, exhausted, markers being exhaustion marker being high. And also we also compare side by side with those 3 donors to give them like a different cell line, U87 glioblastoma cell line, to repeatedly stimulate them. And then we found that the cell decline a lot quicker. So then we do this bulk RNA sequencing at 24 hours after stimulation to see what's going on in terms of downstream signaling when we activate the CAR T cells. Actually, interestingly, between the comparison between the particle stimulation versus the cancer line U87 stimulation, we saw that the pathways, like including DNA repair, mitochondrial function, telomere, and cell cycle related genes, all being up, while the immune activation associated genes or signalings are actually down. So by this, I hypothesize that our stimulation maybe find a sweet spot that we can actually uncouple T cell proliferation from excessive immune activation, which make them can live longer and proliferate massively. The last point of data I want to share is that this is intrigued by other studies on mouse T cells that they found T cells can go supernumerary expansion. But with human cells, we also see supernumerary expansion. Do we see the telomere length shortening? So in the mouse work, the telomere seems to be constant. But in our human T cell data, we found that the telomere is actually shortening. But we saw more of a stable region when the telomere length is stable among these many different donors. So there's some reason mechanismally that we found can partly answer is with particle stimulation, we saw a lot higher level of telomerase activity. So this actually makes sense together that maybe the particle stimulation is giving more of the salary to put in the bank, so that the telomere maintenance is more stable among this massive cell expansion. So, and also I want to point out this stimulation is only temporarily increase the telomerase activity. So as a summary, that we really find an interesting mechanism to uncouple the cell proliferation from excessive activation, and then the cell expansion fold look quite consistent among different donors. This can point to new interesting directions that my lab right now is focusing on. Can we apply this in more like universally applicable in different CAR receptors and TCR receptors? And also we are actually really validating the efficacy in vivo. And also we were interested in applications in challenging T cell manufacturing cases. And due to the time issue, I really want to quickly go through a different direction. As mentioned, that these particles, they are made of biodegradable materials. They can be they can be administered in vivo. So in our published work, we show that working with synthetic biology tools, we can actually locally activate CAR T cells through a synthetic notch receptor engineered CAR T cells. So this concept can be extended to give a multitude of different signal at a precision way pattern to modulate immune cells to help a cancer vaccine-like idea. So this is the direction that my lab is actually actively working on. And the last thing I want to mention is this is a very precision biomaterial platform that we think it may be useful for a lot of immune modulations. And because the technology actually converge very well with a lot of sequencing antibody DNA conjugates has usually been made. So, but to me, I feel like if not a material lab, maybe there's some challenge to get this DNA scaffold particles, and we're actually planning to make and then share those particles. So to collaborators and also to people who have interest using this platform. So please stay tuned at my LinkedIn and Twitter account. So I would definitely like to help moving using this platform to advance the immune modulation. So as I mentioned, this is a work done at UCSF, and there's many collaborators, and I really get a lot of support from my mentors and collaborators listed here. And also, this is my new lab, and with a collaborator working on computational analysis of T cell signals. So that's all, and thank you for your attention.
Great. Thank you so much, Dr. Huang. That was an amazing talk, very complimentary to the first talk that we had today from Dr. Bassi. So really two fantastic talks. And now I think we can start the Q&A session. So if you're holding on to a question, just as a reminder, you can put it in the Q&A box and we'll try to get to it if we have time. Since Chris, since you gave your talk first, let's start with a few questions relating to your research. We have one here from one of the attendees who asks, have you looked to see whether your GPC2 positive EVs also contain packaged RNAs or double-stranded RNA fragments that could be eliciting some immune response? So I guess, is this EV phenomenon that you're observing dependent on GPC2 expression within the EVs, or is it something else?
Yeah, we haven't looked for other components of EVs. There surely are a lot of them, but we thought about this. So we've used paired EVs, so from isogenic cells. So we make put GPC2 into 293 T cells and then capture EVs plus and minus GPC2. So we think it's what we're seeing with the CARs is GPC2 driven, because we've tried to control for other elements that may be doing. And a lot of people have showed PDL1 on EVs being immune modulatory, which makes sense, but we've tried to control for that as best as we could.
Great, thanks. We have another question related to your talk, Chris. The attendee asks, is the GPC2 topology in EVs known? For example, is GPC2 fully encapsulated in the EV? Is there a transmembrane or ectodomain on the surface of the EV? Have you been able to basically characterize GPC2 on EVs versus GPC2 presented on the tumor cells themselves? Yeah,
so what we know is it's extra EV, or it's on the surface. As far as we can tell, we can see it by flow cytometry using the same antibody, actually the antibody we use to flow cells, which is the same antibody that is on the CAR, or the CAR is made from. So it seems to be extracellular. Whether it's the exact same topology, we don't know. I mean, I think given the conformational aspect of the CAR epitope, I presume that it's a similar topology, but it seems to be extracellular. I think what we're learning is what's on the cell is also on EVs secreted by those cells, but whether they're exactly the same conformation, we don't know that yet.
Yeah, definitely a topic for future investigation. Another attendee asks whether you are looking into other targets to overcome GPC downregulation or loss. So in other words, Are you developing ways to target multiple antigens along with GPC2? Is this even a problem in neuroblastoma? Can you talk about your lab's current efforts to work in this space?
I mean, you know some of this work. So, you know, I think with several GPC2 CARs now we've seen when they work really well, you get antigen downregulation, which I think is probably true of every CAR that works well at some level. And so, yes, it's an area of active investigation. And one way, obviously, to circumvent that is dual targeting potentially. So we have several programs focused on that. So more to come for sure.
Great. And then maybe I can ask a brief question about some of your work. To me, in some ways, it seems a little counterintuitive that EVs would enhance CAR T-cell therapy. And when I'm imagining this in my head, I'm thinking that EVs may sterically block the CAR from binding to antigen presented on the tumor and sort of distract them in a way. So is some of this enhancement, do you think it has to do with like bystander killing or some other mechanism that people haven't really thought about in the context of CAR T-cell therapy? Like, can you give us some insight into how these things might be working?
I agree. I think, I don't know, is the answer, but I think there's some literature about GPC3 actually coming out of the NCI that it acts as a sink. So it actually gets cleaved and acts as a sink for antibody therapies, right? So getting at your same point, you would think that this would distract T cells. So I don't know. I mean, we're just getting into this and it seems to enhance the efficacy, but whether you have a CAR T cell that interacts with the EV, then releases the EV, but is still activated and then is able to, you know, or is it just, does it just proliferate and then these, you know, you just get more of a mass of T cells maybe is what's happening just because you have this extra tumoral sort of signal. It's the way I sort of think about it. And maybe it just solves, it gives, you know, it's like CD19, right? It gives you antigen that's readily accessible and then you just get proliferation. And then, but I would think that once you encounter EV, that T cell is kind of done. And maybe there's enough stimulus to just get more, but you know, it's a good question. I think, you know, it surprised us too. And there's a lot of literature showing the exact opposite. And a lot of that's probably driven more by PD-L1, to be honest with you. So I think, you know, we're maybe just lucky in that sense that we don't have inhibitory receptors on our EVs.
Yeah. I mean, this also, we know that like CAR T cells need to reach a certain activation threshold to fire. And if there are GPC2 low cell, you know, neuroblastoma cells that CAR T cells can't recognize all that well, maybe the EVs sort of help rev them up and just enough to kill. But be looking forward to understanding more about this really interesting phenomenon. So thanks, Chris. Maybe we'll move on to a few questions for Dr. Huang's talk. So one of the attendees asks, with the repeated ICEP stimulation, do you see increased telomerase activity at the 120-day time point, in addition to the time points that you showed in your talk?
That's a great question. Actually, I would say due to the time I go very quickly about the slides. So what we saw is within the first 3 to 5 rounds of stimulation, that's when we see a lot augmented telomerase activity. And then after that, it's actually going down. It's compared to non-stimulation or compared to other stimulation, it's not as dramatic in enhancement. So this is also, to me, it's maybe a good thing because it's more physiological level. We're not actually getting cancers like cell features. So yeah, that's
Great. Yeah, thanks for that. Let's see, another question about ICEP polymer composition. So one of the attendees is wondering whether these polymers could be changing the metabolism of CAR T cells to enhance metabolic fitness, and sort of whether this relates to memory T cell differentiation or not. And did you find any metabolism pathways in your transcriptomic analyses? You mentioned some sort of mitochondrial related pathways, but can you give us some insight, maybe a little more insight into the metabolic profile of these cells?
Yeah, so from our analysis so far, we just see like the mitochondria associated pathways are augmented. So, and then functionally through seahorse, we see enhanced mitochondrial fitness.
Mhm.
So, how did this get into the signaling there? That's something we're not sure yet.
Right.
Yeah.
Right. And it's interesting because it seems like maybe at least a portion of your cells maintain some sort of effector memory or memory-like phenotype, which is associated with OXPHOS metabolism. So it's interesting that you may be able to sort of occupy, but maybe both phenotypes simultaneously, a cell that's sort of activated and dividing, but also maintains proliferative potential.
Yeah, related with this comment, so there's another side of the data I forgot to mention is we actually look at during our S particle activation within an hour to do seahorse analysis of glycolysis versus OXPHOS. Actually, interestingly, compare with a cancer cell stimulation, it's lower for OXPHOS compared to cancer cell stimulation. But glycolysis is, I would say, not as dramatically lower. It's somewhere lower, but not as dramatic lower as OXPHOS pathways. So this we're still trying to digest and interpret what does this mean and how is this connected to their cell phenotype.
Right. Right. One of the things that we see in the lab that relates to this work is, when you have a synthetic cell, sometimes you get synthetic phenotypes that don't really match dogma in the field. And it certainly seems like this is something you're running into as well.
Yeah, totally. Yeah.
Great. So another question from an attendee, Dr. Huang, thank you for your great work. Do you envision an implantable ICEP in the future for in situ or in vivo reprogramming of T cells to basically overcome exhaustion and other challenges in the field?
Yeah, that's exactly what we're working on right now. So there's challenges if we give the biomaterials in vivo in terms of what is their biodistribution and how long they can be stable in vivo. So actually with that, we're more excited about in situ or local administration. So because we believe that during for solid tumor after surgery, there's maybe a great time frame that we can use functionalized biomaterials to actually first help T cells, and also secondly, if we can engage innate immunity to be more generate systemic immunity, that would be really fantastic. So that's definitely the direction we're really excited about.
Well, we look forward to seeing those studies emerge from your lab over the next few years. Maybe one last question, Dr. Huang. So, I was really interested in your work sort of as a model of T cell proliferation or exhaustion. But I'm wondering, you kind of alluding to the translational potential of these polymers and biomaterials in terms of changing CAR T manufacturing and enhancing fitness. So I'm wondering whether you're thinking about this in a translational way as it pertains to manufacturing. Are these cells receiving some sort of beneficial signal that's different, fundamentally different from, let's say activation beads or anti-CD3 or traditional methods to expand T cells in culture for clinical manufacturing? Or is this really just a way to get more insight into how human engineered T cells expand and differentiate?
That's a great question. So I would say my interest goes on both. First, as for manufacturing, we're not interested in like culture them for 100 days. That's too much high cost. So we're actually excited about can we actually enrich those long-lived clones with some rounds of stimulation to get a better product. That's for manufacturing. So yeah. So on the other side, in terms of a model, so I think this is also maybe a great model to study T cells for such a long time. For example, how exhaustion is mechanistically decided with the signal we give, and at a certain point you can decide to give we actually right now know some signal that is making T cell exhausted quickly, while some others make them live longer. So this this is something I actually really excited for some collaboration on epigenetic part of the T cells, which definitely your work is quite closely related. Yeah.
It's great. Now, I think we'll learn a lot from these studies, and just the feat of culturing human T cells for 120 days, I think deserves deserves some credit, because that's a lot longer than most people are willing to take out their in vitro experiments. So, well, it looks like we've answered all the questions. And
Just one last question.
yeah.
I want you to knock that out.
Oh, sorry. Oh, this one,
yes.
Dr. Bossi. Yeah, we sort of
Okay.
addressed this a little bit.
Okay.
The question was about how stable the expression of GPC2 is during the development of the tumor. So, Chris, we talked about GPC2 expression in the context of a CAR T cell response, but how about in terms of the development of these tumors?
Yeah, I mean, as far as we know, it's stable. It's important. You knock it down, the tumors don't tolerate it well. I think it's in all different stages. So I think it's important, but it's a hard thing to really study. But it seems to be important in development, important in tumors, and not in between. So I think, yeah, that's what we know.
So with that, I'd like to thank Dr. Bossi and Wang for their excellent presentations. Thank you, Dr. Weber, for your excellent moderation. Our next Cancer Moonshot seminar series coming up is Implementation Science and Patient Engagement Approaches to Improve the Early Detection of Treatment in Cancers, Dr. Saka and Dr. Tomasoni Ardori. And with that concludes our today's session. Thank you very much.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.